Good afternoon, everyone, and welcome to the ICTS Precision Medicine session at Venture Café. I'm your host, Rob Culverhouse, Associate Professor of Medicine at WashU. We meet on the third Thursday of the month at 4 p.m., and these virtual sessions last 50 minutes. As always, our mission is to connect innovative biomedical science generated at WashU, SLU, and MU with entrepreneurs and businesses in the St. Louis region. We hope these sessions stimulate your thinking, and we encourage you to share your insights and ideas with the speakers after their presentations. I'll mute the audience at the start of the presentations, but feel free to unmute yourself when you have a question. Today's presentation is on an issue that has likely touched everyone here, translating genetic insights into treatments for Alzheimer's disease. Our speakers are Drs. David Holtzman and Marco Colonna. Dr. Holtzman is Professor and Chair of Neurology at WashU, Scientific Director of the Hope Center for Neurological Disorders, and Associate Director of the NITE Alzheimer's Disease Research Center. David has focused much of his efforts over the last 25 years on trying to better understand the mechanisms underlying neurodegeneration, particularly as they are relevant to Alzheimer's disease. Over the past 6 years, Dr. Holtzman's lab has been studying how microglia and genes such as TREM2, which Marco will talk about, influence neurodegeneration. Dr. Colonna holds the Robert Rock-Belliveau MD Professorship of Pathology at WashU. His lab is broadly interested in innate immunity, and one of the main areas he focuses on is innate immune mechanisms in Alzheimer's disease and neurodegeneration. Today, he'll tell us about the role of the gene TREM2 as a possible treatment target for Alzheimer's disease. So thank you both for being with us. And David will start us off with a presentation on ApoE, the best known gene related to Alzheimer's disease.
Well, thanks for the introduction, Rob. And I think it's good that Marco and I will both speak at the same session, and I'll try to say anything that I do that is and not overlap with what I think he'll probably talk about. Given the audience for this and purpose for the Venture Cafe, I'll try to illustrate why ApoE is important for Alzheimer's disease, but also it's not been up to date been a major target for therapeutic intervention, but I do think it holds a lot of promise as a target, and so that's what I'll try to illustrate in my talk. Let me share my screen. Everybody should be able to see that okay. Here are some disclosures. What I'm going to talk about today is related to ApoE, which is, and I do have one relevant disclosure with that, which is that we've produced some anti-ApoE antibodies that have been licensed to a company, and then we are working on a grant related to that topic. So as all of you are probably aware, dementia is a decline in memory and thinking that leads to social and occupational problems. And Alzheimer's disease is one of the diseases that can cause or contribute to dementia. In the United States, it probably contributes to about 70% of cases. We know a lot about the underlying pathology that is associated with the disease. It was described over 100 years ago. And what you find in the brain, as illustrated in this panel on the right, is the accumulation outside of cells of these things called amyloid plaques, which are made up of the amyloid beta peptide. And you also see, as shown in this microscopic slide, the accumulation of what are called neurofibrillary tangles, which are made up of a protein called tau that is, in both cases, these proteins that accumulate are normal proteins, but they clump together and aggregate, and it's believed that the buildup and aggregation of these proteins is important in the causation of the disease. Like every other neurodegenerative disease, in addition to this protein pathology, you also have loss of nerve cells and their connections, synapses. You also have a strong inflammatory response that I'll touch on a little bit, and I'm sure Dr. Colonna will touch on more. One of the important things to know about Alzheimer's disease is that when people develop the symptoms, as shown on the slide, where it says very mild Alzheimer's disease or mild cognitive impairment or very mild dementia. That's the very earliest clinically detectable stage of the disease, but that's not the beginning of the disease. It's by the time you're actually have any symptoms, even very mild, the accumulation of that protein I just showed you called amyloid is already pretty much peaked in the brain. And the accumulation of this neurofibrillary tangles, or tau, here in blue, is on its way up. It's not at its peak yet, but it's already there. And the amyloid pathology has been shown to start building up about 20 years before the symptoms build up. And I think this is a really important thing that's been discovered. Much of the work discovering this was at our Alzheimer's Disease Research Center. And it's been using a variety of different participants who've been very kind to volunteer to study these changes over time. So ApoE is the strongest genetic risk factor for Alzheimer's disease. And as you can see in this graph on the left, there are 3 different flavors of ApoE. ApoE is a gene that encodes a protein that's 299 amino acids, and at position 112 or 158, there's literally just a single amino acid change that results in a huge change in risk. So in this graph, if you're unfortunate enough to have 2 E4 alleles, the average age of onset of dementia in this particular population was about age 70. Whereas if you had 1 copy of the ApoE2 allele, your average age of onset dementia was well over 85. And so just to put numbers on this, if you have 1 copy of ApoE4 versus what we call the neutral copy of ApoE3, your risk is about 3.7 times higher to get Alzheimer's disease. If you have 2 copies of ApoE4, your risk for Alzheimer's disease is about 12-fold higher than somebody who's E3/3. And E2 decreases your risk by about 40 to 50%. So this is a huge effect on disease risk. And as I mentioned, this protein's 290 amino acids. It's produced at very high levels in the liver, but also very high levels in the brain. Where in the brain, it's made mostly by glial cells, astrocytes, and also when there's damage in the brain, it's made by microglial cells. It's interesting protein because it's in lipoprotein particles that in the periphery control cholesterol and lipid metabolism, although in the brain, it's not entirely clear the normal function of ApoE. One of the disease-related functions is that it actually binds to all proteins that form amyloid, including the amyloid beta protein. What's been shown to date in a series of many different studies in both animals and humans is that ApoE influences several different stages of the pathogenesis of Alzheimer's disease. It influences the accumulation of amyloid plaques, it influences the inflammatory response, and it influences the phase of disease where tau is accumulating and the brain is degenerating. So I'll just briefly show you this information next. So if you take an animal model that develops amyloid accumulation as shown in the top, just like occurs in humans, and you simply cross in the disease risk factor ApoE4 versus the neutral form ApoE3 versus the protective form ApoE2, you see this dramatic difference in the amount of amyloid that builds up in the brain. And this is very similar to what you see in humans. One question would be, so, okay, well, if that's true, if from a therapeutic perspective, would you want to decrease or increase this protein? And in genetic experiments, if you do an experiment where you simply have an animal that has 2 copies of E3 or 2 copies of E4 that also develops amyloid deposition, you can see if you lower the amount of this protein, ApoE3 or 4, by literally 50%, you dramatically decrease the accumulation of this amyloid protein, which we think is a necessary component in the development of Alzheimer's disease. So what about ApoE's function in regard to the glial cells, and why might that be also relevant? So in a series of studies that we've recently done in models that develop tau pathology, these are brains of mice that develop tau pathology, and they develop clear-cut neurodegeneration with loss of brain tissue and loss of neurons in regions of the brain I'm circling here, like the hippocampus. If you compare this hippocampus of a mouse, which looks very similar to that in a human, to a normal mouse, you can see that it's dramatically shrunken. And these mice express tau pathology and also ApoE4. And you can see when these tau mice express the ApoE3, the neutral form of ApoE, or the protective form E2, they have much less damage in the brain. And if these mice express no ApoE, there's literally almost no damage in the brain at all. So one of the things that we found is that if you look at genes in these mice that have tau pathology and express ApoE4, there's a strong increase in genes in the microglial cells, the inflammatory cells in the brain, that are increased in the mice that express E4 in red versus the ones that don't express ApoE. And if you look at staining of the brains with microglial markers that show activation states, you can see that the E4 mice have a lot more what we would call activated microglia. And I won't go through a number of studies that have suggested now that somehow we have evidence now that ApoE4 in particular is driving the microglial inflammatory response in the setting of neurodegeneration. So this is dramatically illustrated in these experiments here, where if you look on the left, these are these tau E4 mice that develop a lot of damage in the brain with shrunken hippocampus, shrunken cortical regions. And if you simply remove the microglia with a drug that gets rid of the microglia from the brain, we can block all of the neurodegeneration that occurs in these animals. So we think that there's now evidence that ApoE, through somehow acting through microglia, is driving the damage in the brain that we see in a model of degeneration in Alzheimer's disease. So in summary, we know now that ApoE strongly affects amyloid deposition in the brain. So that's the first pathology that builds up in Alzheimer's disease. And it appears to do so by affecting the clearance and seeding or buildup of this protein. And it's likely one of the major reasons why ApoE is linked with risk for this disease. But in addition, ApoE4 in our model systems increases neurodegeneration phase, or the damage phase, that seems to mimic what happens in Alzheimer's disease. And the absence of ApoE, or more recently, what I don't show here is lowering ApoE in a brain, seems to be protective. And I think these effects appear to be somehow mediated by the microglial cells that Dr. Colonna will talk about more. There are other effects I'm not going to talk about here that are pleiotropic that ApoE may play on other issues, such as the blood-brain barrier, synaptic plasticity, etc. But we don't know if these other roles that ApoE has on Alzheimer's disease we don't know if these other roles contribute to Alzheimer's disease or not. And finally, I think this is what I wanted to discuss after I end my talk in a moment, is from a therapeutic perspective, it would appear that at least for these ApoE4 and ApoE3, the most common forms of ApoE present in humans, that decreasing the levels of these proteins would be promising, would be a promising therapeutic target. So what we do with ApoE2, which is less common, is not clear, but I think a significant challenge going forward is how do we lower these proteins to potentially get a therapeutic effect and turn it into some sort of a therapy. So initial thoughts might be things like gene therapy or other techniques that we can further discuss. I mean, you're probably aware that there are ways now to lower proteins in genes and proteins in the brain with techniques like antisense oligonucleotides. Just in the last 2 years, this approach has been used in a terrible neurodegenerative disease called spinal muscular atrophy, where children usually die within 1 to 2 years of life. And when this approach using an antisense oligonucleotide for another gene is given, these kids now survive and if started early enough, actually can be pretty normal. So that's one possibility, but gene therapy would be another possibility. But maybe there's others as well. So I'll just show a picture of the many people that contributed to the work I showed today. So I'm happy to have discussion further at this point. I don't know how you guys run the discussion, so.
Yeah, so when people have a question, just unmute yourself and go ahead and ask. But I'll start you off. I just wondered if, well, a couple of things. On the mice that you have done knockouts on and stuff, are they otherwise normal, or do they have all sorts of problems, either from knocking down or knocking out?
Yeah, so ApoE knockout mice are pretty normal in terms of their brain function. There's not a major problem. But when you knock out ApoE peripherally completely, it markedly alters cholesterol metabolism. That the animals or humans that have, rare humans have a complete defect in ApoE as well. And they have very elevated levels of triglycerides and large lipoprotein particles. So you wouldn't want to lower ApoE in the periphery to very low levels. You could lower it down by 70–80% without having an issue, but you'd probably want to target the lowering for the central nervous system as best you could. You wouldn't want to go too low peripherally.
Okay, and is that related? I believe I thought that ApoE2 did have some increased risk for heart disease, maybe. Was that cholesterol related?
Well, it's if you only have one allele of ApoE2, you actually ApoE4 is linked with a somewhat of an elevated risk for heart disease. ApoE2, when it's a very rare allele, and it's present usually in people if they have it in one copy, and in that case, it's not a risk for heart disease.
And then the other thing, real quickly, is what about should we, once we get a prophylactic treatment, should we all be starting it early, or is there a way to detect that start of plaque buildup?
Yeah, there are actually, that's a good question. There's a lot of ways to now detect the accumulation of Alzheimer's pathology in that period that leads to that 20-year window, or even before it. Many of these are not approved, are not are done in our research studies and are done in clinical trials, but insurance at this point won't pay for them. But once there are in trials, but also as therapies are developed, there are great ways that can detect these things, both with imaging of the brain with different types of agents, with cerebrospinal fluid studies, and more recently now, some of these changes can be seen in blood with blood biomarkers. So yeah, I think the best way ultimately to treat Alzheimer's disease will be to prevent or delay it, and we're going to need to use biomarkers and start assessing people in their middle age period to begin to assess what's going on. Kind of like what happens in heart disease, where you're assess risks beginning, after age 20, 30, and start seeing whether different things start changing or not. And then if they do, to treat them.
David, I have a question. ApoE obviously is an important molecule in lipid metabolism. So I was wondering whether there's any connection between the neurodegenerative phenotype that you see and the lipid metabolism in the brain. Yeah.
That's something that we've been really interested in for a long time, and are in the midst of some studies to work on that. So if you completely remove ApoE from the brain, and you measure different levels of cholesterol metabolites, phospholipids, you really don't see any change. But in the last few years, what people have started to see is that if you look at specific cell types in the complete absence of ApoE in the brain, then you can start seeing that there are changes in cholesterol esters that accumulate, particularly in microglia and to some extent in astrocytes, but not in neurons. And in the setting of neurodegeneration, there's also a similar accumulation of cholesterol esters in microglia and a few other phospholipids. So what we're trying to figure out now is if we alter ApoE levels or its lipidation state, whether that would alleviate the cholesterol and phospholipid accumulation that occurs in, for example, in microglia, and maybe that would be a way to decrease some of the inflammatory changes that are occurring in this setting.
Okay, thank you.
So it might get rid of cholesterol from microglia in particular. That would be the hypothesis.
Hi, Dave. I have a question about ApoE2 again. I haven't given it much thought, but has anyone tried somehow, raising ApoE2 levels in mice and seeing if, that is actually as protective as you would think, or is it just that
Right.
ApoE2 is not as expressed or something in the models? I was
Yeah.
wondering about that sort of as a way to target it.
Yeah. No, that's a great question. It's ApoE2 is expressed at the same level in terms of the gene expression in both humans and the animal models, but in some studies in animal models of amyloid deposition, there have been a few papers where people have overexpressed ApoE2 in the brain. And in general, it seems to decrease, in the case of amyloid, it tends to decrease amyloid deposition to some extent. The studies haven't been done, in my opinion, in the correct way yet, but what hasn't been studied yet is if you overexpress ApoE2 in the brain, how it affects some of these other parameters like the inflammatory response or tau or other things. So I think there's a lot needs to be done because clearly it's a very having it present throughout your whole life is protective. So. Some of the proteins that regulate ApoE levels normally are one is one of the most studied proteins in biology, which is the LDL receptor, which is the main receptor regulating ApoE levels in the body. So that could be a potential target to influence ApoE in the brain.
Yeah, I was wondering, of these, I guess, therapies or therapeutic techniques that you've mentioned, what you think about things that need to cross the blood-brain barrier and what is going to be safe in addition to being effective.
Well, one of the ways that's been used recently in the spinal muscular atrophy and with these antisense oligonucleotides to affect gene expression, you have to administer them in the CSF. But the next therapy that was approved soon after it was an adeno-associated virus that's delivered peripherally. And enough of this particular AAV vector in humans gets into the CNS to express the SMN gene product to essentially cure SMN if given early as well. So there you're giving that particular AAV gene therapy peripherally, and enough gets in the brain to have a very strong effect. That was developed by a biotech company and sold just a few years ago to Avexis and Novartis, or well, the company was Avexis, but it was sold to Novartis, and that's now the most expensive single gene therapy for any human disease. But it works. The bottom line is it works.
Right. Well, thank you.
In the CNS, it works in the CNS, even though it's given peripherally. So.
Is that, I mean, do AAVs generally get into the CNS? No problem. And is the problem more of the cell type that it's affecting?
And not all AAVs given peripherally get into the CNS. It all depends on the capsid that's used, and the so it's. I think that's an area that needs to be worked out a lot better. But there are a lot of companies are working on this now.
Cool. Thanks.
Rob, your microphone isn't on.
So thanks, and we can always circle around again at the end if there are more questions for David later, but let's now welcome Marco, who'll tell us about the gene TREM2 and Alzheimer's.
Okay. Let me just share the screen. Can you see my screen?
Yes.
Okay, great. So I'd like to tell you about our effort to exploit TREM2 in the therapy of Alzheimer disease. These are my disclosures. We became interested in Alzheimer disease fairly recently because of the genetic studies linking DNA polymorphism associated with Alzheimer disease with genes that are expressed in microglia. For example, MHC class 2 or SPY1. And we heard from David that ApoE is also expressed by microglia. But we were particularly interested in this gene, TREM2. In some rare individuals, the mutation R47H or TREM2 is associated with high risk to develop Alzheimer disease. And we were interested in this receptor because we actually cloned it many years ago. It's a cell surface receptor of immunoglobulin superfamily. And it has one single immunoglobulin superfamily domain. Has been shown by us and by others to bind to lipids, particularly phospholipids, which can be exposed on apoptotic cells, but can also be found in lipoprotein like HDL, LDL, and ApoE. Now, this receptor does not signal on its own, but needs to associate with a separate signal transduction subunit, which is called DAP12, which is responsible for signaling. So when we look at brain section of mice that accumulate A beta, for example, this mouse model of Alzheimer disease called 5-XFAD, we see that plaques in greens here are completely surrounded by microglia that forms a barrier that prevents the spreading of the plaque and eventually neurotoxicity of a plaque. But when we cross these mice on a TREM2 deficient background, we found, actually in collaboration with David, and particularly with Jason Orrick, that there was a lack of clustering of microglia around A beta plaques. And this, of course, has consequences on the plaques, because when you have microglia around the plaques, plaques are more compact. But in a TREM2 deficient background, where you have very little microglia around the plaques, the plaques are more diffuse and tend to spread into the parenchyma, and they're more damaging. And in fact, if we measure neurite dystrophy around the plaques by staining brain section with for N-terminus APP, we see much more neurite dystrophy around the plaques in a TREM2 deficient 5-XFAD mice than in a 5-XFAD mice. Now, we were interested in understanding the mechanism by which TREM2 controls microglia clustering and ability to contain plaques. And so for this, we collaborated with Ido Amit at the Weizmann Institute, and we performed single cell RNA-seq. So essentially, what we do here, we isolate microglia from the brain and then define the transcriptome for each single cells. And then computationally, we can define different clusters of microglia cells based on their transcriptome. And what Ido have found is that the majority of microglia form a cluster that we call homeostatic cluster. But in aging mice, some microglia form a separate cluster that expresses a number of activation markers. For example, TREM2, but also molecules involved in lipid metabolism, for example, ApoE, molecules involved in inflammation like SPP1, molecules involved in lysosomal biology, for example, cathepsin D, cathepsin B. So when we look at wild-type mice in steady state, we see that the majority of microglia form these homeostatic clusters. In a 5-XFAD mice, where we have accumulation of A beta plaques, we see that some microglia cells form this cluster of activity cells that we call disease-associated microglia. And in a TREM2 deficient mice, we see the microglia cannot become completely activated, and the majority of microglia actually partially activated or non-activated. So we actually distinguish two steps in microglia activation. One is TREM2 independent and one is TREM2 dependent. The majority of the cells in a TREM2 deficient mice are mostly partially activated or non-activated. Now, I've taught you about I've shown you the TREM2 deficiency, but as I mentioned, individuals with a risk for Alzheimer's disease have a single point mutation of TREM2. And so we wanted to establish the impact of this variant on microglia activation. So we generated transgenic mice that expressed either a common variant, human variant of TREM2, or the R47H variant of TREM2, which is associated with Alzheimer's disease. And then we look at the clustering of microglia around plaques. So when mice express a common variant, we see a nice cluster of microglia around the plaques as compared to the knockout mice that do not express any TREM2. And when we look at mice that have the R47H variants of human TREM2, we see some limited clustering of microglia around the plaques. So there's the quantitative effect of clustering. So this experiment really tells us that this variant of TREM2 is reducing microglia response to pathology. This defect is a subtle defect, but we speculate that accumulation of this defect over the lifespan of an individual can actually facilitate the progression to Alzheimer's disease. Now, the microglia defect is probably not the primary cause of Alzheimer's disease, but accelerates disease progression. And of course, you need additional factors like age, genetic factors, environmental factors that will contribute to Alzheimer's disease. But most importantly, this experiment told us that perhaps if we can activate TREM2, we may activate microglia and perhaps have some therapeutic effect on Alzheimer's disease. And so to test this hypothesis, we collaborated with a company, Alector, that has developed a TREM2 antibody for clinical purposes. And this antibody has already been tested in phase 1 clinical trial for Alzheimer's disease. And the work I'm going to show you was done by Shutang Wang in the lab. So we obtained this antibody, AL002C, which binds to both the common variant and the R47H variant of TREM2. We demonstrated that this antibody is actually an agonistic antibody by using this reporter assay, in which engagement of TREM2 leads to expression of GFP. And as you can see in this assay, the antibody activates the common variant and also activates the R47H variant of TREM2. And now we tested this antibody in our humanized mouse model, in which mice express either a common variant or the R47H variant of TREM2. We did one assay in which we injected a single dose of the antibody intraperitoneally, and then we look at microglia activation by single-cell RNA-seq. Now, when we do single-cell RNA-seq, we see homeostatic microglia, we see activated microglia, or DAM, but we also see interferon imprinted microglia, microglia that's duplicating the DNA, microglia in the M phase, and also metabolically active microglia. So we look at the representation of all these clusters after a single injection of the antibody. And what you can see is that the antibody is inducing the expansion of only two subsets of microglia, metabolically active and the proliferating microglia. Now, we also tried a chronic treatment of these mice with an anti-TREM2 antibody to see whether the antibody has any impact on the pathology in these mice. As I mentioned, microglia activation limits the spreading of plaques. And so in the presence of microglia, plaques are more compact, and in the absence of microglia, plaques are more filamentous. And we can distinguish filamentous from compact plaques using different stainings, 6010 staining or methoxy XO4 staining. But the bottom line is that when we look at filamentous plaque that we can visualize with the antibody, but not with methoxy-4, we see a reduction of filamentous plaques in mice that have been treated with the antibody. When we look at inert plaques that can be visualized by methoxy-4, but not by 6010 antibody, we see an increase of inert plaques. And also we see that what we call compact plaques are basically unchanged by the treatment of antibodies. So essentially, it appears that injection of antibody reduces the neurotoxic activity of the plaques. And when we measure dystrophic neurites around the plaques, indeed we can see that in both CV and R47H transgenic mice, there's a number of neurite dystrophy around the plaques. But when we treat the mice with the antibody, we see a reduction, a significant reduction of neurite dystrophy around the plaques. So in summary, what I've shown you is that a single injection of an anti-TREM2 agonistic antibody in a mouse model, humanized model that expresses human TREM2, expands microglia, a subset that proliferate and are metabolically active. And that a chronic administration for about 3 months of antibodies reducing filamentous plaques and reducing neural damage. Now, an experiment that I didn't have time to show you, that were performed by Calagiere on the behavior of these mice, we found significant changes in some behavioral tests in mice treated with antibody, particularly in risk-taking and anxiety-like traits. And again, in data that I didn't have time to show you, we found that indeed the antibody is inducing microglia activation and particularly increased activity and increased phagocytic activity. So essentially, we think that this experiment show or provide some evidence, supporting evidence that antibody engagement of TREM2 can increase neuroprotective microglia, can reduce toxicity of A-beta plaques, and therefore can be beneficial in Alzheimer's disease. So this is my lab. I mentioned Xu Tong, who did this work, but I like also to mention Ingrid Zhu, Yun Chen, Jin-Chao Hu, and Vincent Pegg, who are working on this project together with Marina Schell and Susan Gilfillan. And I like to mention the Alector team, which helped us with the characterization of the antibody. Of course, we collaborate with David and Jason, with Max Artyomov, and with Ido Amit for single-cell RNA-seq. And again, I like to particularly mention Calagiere for the testing the behavior of these mice. So I will stop here and I'll take any question.
So thanks, Marco. I saw that you had, you said that you'd tried or started a phase 1 trial in humans. And so do you have any idea of whether or when that will progress to future stages?
Yes, so the phase 1 clinical trial showed that the antibody is safe. And it also identified 2 potential readouts for target engagement. And one interesting is a soluble CSF1 receptor, which can be detected in the cerebrospinal fluid. CSF1 receptor is expressed by microglia, and therefore we speculate that engagement of microglia and increased proliferation of microglia will actually facilitate also the release of the increased expression of CSF1 receptor and the release of soluble CSF1 receptor. The phase 2 clinical trial will start in the fall, and I guess we'll this is not, I'm not directly involved in this. You know, this is driven by Alector, but there's additional companies that have developed antibody anti-TREM2, and I'm sure that they will be testing as well.
Great. I guess also you were talking about at the end about the prolonged exposure to the treatment in mice. And so it sounded like this was actually rescuing already damaged mice, which it seemed like there's been some speculation that some of the treatments are only good or might only be good before the damage has occurred. But
yeah, we deliberately started the treatment at 5 months old in these mice. At that age, mice already have developed plaques. and they have already developed a microglia reaction. So we wanted to test whether the antibody may have a therapeutic effect, and that seems to be the case. I have to point out that the effect is not dramatic, but is statistically significant. We have repeated it. And we actually have another collaboration in which we have obtained similar results with using a different antibody. So we're pretty confident that the antibody will have an effect also in a chronic treatment. Of course, these are mouse experiments, right? So in human, of course, things are different because the mouse, one of the limitations, of course, is the mouse model is essentially a mouse model of A beta accumulation. There's no tauopathy here. And when we look at human Alzheimer disease, especially in advanced phase, you just don't have A beta accumulation, but you also have tauopathy, you have neuronal degeneration, neuronal loss, synapse loss. And so it's difficult to predict exactly what the antibody treatment will do in those conditions.
But I was interested that you said that you could detect changes in behavior, and it wasn't just the biochemical changes that you could detect.
Absolutely. And I have to say that these changes in behavior are also very robust. They're not dramatic changes. Again, they're subtle changes, but they're very reproducible. And you know, these were done especially in a couple of tests that measure anxiety in mice. So usually anxiety is in an elevated plus maze test. We measure the time that mice spent in open arms versus closed arms. And when you have A beta accumulation, mice spend more times in open arms. And when we treat the mice spend less time in open arms and more time in closed arms. So they seem to become wiser.
Great. Anybody else?
I have a question, Marco. Do you know is anybody trying to develop small molecules to affect TREM2?
Yes. There's a list, a couple of companies that we know they're developing small molecules that target TREM2. And we also have a collaboration with the Baylor College in Houston to develop small molecules that bind TREM2 as well. I have to say that the antibody that we injected, was injected peripherally, but the amount of antibody actually reaches the blood-brain, that crosses the blood-brain barrier is a minimal amount. Right. So usually it's one molecule in 1,000. And so a small molecule obviously will be a big advantage. But of course, you know, there's also more complication related to the potential toxicity of small drugs, right?
Right. Thanks.
Hi, Marco. Could I ask about sort of the, I guess, timing of the way that you can manipulate TREM2, in either direction? If you can activate it, do you know anything about how the microglia respond over time? Does it take time to develop their phenotype, and does that remain stable for a long time, or, you know, vice versa? If you knock it out, do they?
Yeah, well, all we can say at this time is that when we inject the antibody and we look at microglia activation, we see microglia activation already 48 hours after injection. So the effect is very rapid. I can't tell you how sustained is the effect because we haven't done single cell RNA-seq at different time points. We're actually planning to do that. And we haven't done single cell RNA-seq after chronic treatment, which is also ongoing. So the effect seems to be very fast, but we don't know what is the long-term effect of this activation. We don't know if the activation will be sustained, or if the activation will wind down, or if maybe excessive activation may also lead to some sort of energy or reduced activation. So these are different important questions, I think, to address.
Thanks.
Hey, Michael, can I ask a question?
Of course.
So have you looked at neuronal functions, like after the treatment of the antibody?
No, we haven't looked at neuronal function. The only thing that we've done is to do some behavioral studies. And that's the only evidence that somehow when the antibody has an effect on neuronal activity. But we don't have any direct evidence that the antibody has a neuronal effect. I think certainly that would be an interesting question, and we'd be happy to collaborate with anybody to do this, because this is not part of our armamentarium.
Thank you.
Marco, you said only a small amount of the antibody got through the blood-brain barrier. So what expected effects are there on the rest of the body?
Well, we didn't see much of an effect. We didn't see any inflammation or anything like that. But of course, we have to address that more extensively. We really haven't done any biochemical cellular analysis of macrophages in the rest of the body. We know that some macrophages express TREM2. For example, adipose tissue macrophages express TREM2. Other macrophages can express TREM2. And we actually published recently a paper in which we show that tumor macrophages express TREM2 and can be modulated by injecting anti-TREM2 antibody. So certainly there'll be some peripheral effect. They don't appear to be toxic effect, but they certainly could have some impact, especially in long-term treatment.
Well, if people don't have any other questions right at the moment, we're coming right up on the end of our time. So I wanted to thank our speakers again. And
Thank you.
For people who want to follow up later, we often have shy attendees. And so you can visit our website to find contact information for both our speakers tonight. So thank you once again for very interesting presentations.
No, thank you. My pleasure.